Welcome everyone. Today I will be going through an overview of a cloud-based Dockerized and microbiome analysis of biofilm metagenomics. So as a brief background on microbes, microbes are found all around us, in our body and in the food we eat. They actually used to be considered harmful until it was discovered that they can have numerous health and environmental benefits. So biofilm formation is done in 5 stages. A motility stage, which is when the organisms approach the surface. An adhesion phase, which is when the organisms stick to a surface. A maturation phase, when the organisms stuck to that surface will come together to form a complex task. The dispersion phase, which is the formation and it releases balance of the organism. And the propagation stage, which is when they start to begin a new biofilm cycle. So what are metagenomics? Metagenomics is a powerful tool for genomic analysis of biofilm through different sequence identifications. The metagenomic sequencing provides ability to comprehensively sample all the genes and all the organisms present in each biofilm sample. Metagenomic analysis has been really beneficial for the aggregation of knowledge about genes, microbial communities, and its metabolic pathways. So right here we can see the metagenomic process. We start with a community sample of different microbes, and we're really trying to answer the question, who is there and what are they doing? This is done doing different DNA sequencing. There's a 16S-based approach, and there's also a shotgun metagenomic approach. Right there in the middle, you can see what a sequencing machine actually looks like. After this DNA sequencing, the data is put through a quality control process, and I will talk about that a little bit later. And then after the data is analyzed, we can make different discoveries like the questions we were trying to answer. Who is there and what are they doing? So our biofilm metagenomics workflow self-learning module has been designed to understand biofilm's role in plant and human health, which also includes the analysis of the biofilm composition, diversity, and function, and we leverage quorum sensing signature so we can get insights into the microbial biofilm phenotype. The objectives of this workflow are shown right here. The workflow consists of the analysis of biofilm composition, diversity, and function. It's also to characterize the taxonomic diversity of biofilm communities. It's to provide insight into microbial diversity, and it's also for gene prediction. So right here, we can see an overview of what this workflow looks like. Starting in the top left, we can see we have a raw data set. This is put through that quality control or fast QC, which I mentioned earlier, and I will touch on later as well. And then also Trim-O-Matic to fix the data. It is put through an analysis process using QIIME2 and PyCrust. And then using different databases, we are able to do analysis on our data, and we are given an analysis report, which tells us all the things shown on the left side, like what the gene protein, taxa, pathways, network, all that information. So starting with the FastQC and Trim-O-Matic process, right here, there's that picture that was shown on a previous slide of what the FastQC actually looks like, and this gives you information about your data and tells you if it is good to use or not. And then Trim-O-Matic is used to trim out the data that cannot be used to ensure that you have quality data for this process. Next, let's talk about the analysis using QIIME2 and PyCrust2. So when you're analyzing targeted microbiome sequencing, a different approach to determining the origin of sequence is required than typical alignment-based methods. And this is because the gene of origin for an amplified target gene is already known, but the goal is to determine its taxonomic origin based on a potentially small number of variations relative to similar taxa. In the context of whole genome sequencing, A small number of single nucleotide variants, or SNVs, caused by sequencer error are unlikely to seriously confound an aligner and have little effect on the final attribution of the sequence. So in order to minimize the risk of a sequencer error in targeted sequencing, clustering approaches were initially developed. Next, let's talk about the microbe analysis R using the Microbe Analyst database. So this is Microbe Analyst. It gives you a comprehensive statistical, visual, and meta-analysis of microbiome data. It uses marker data profiling, shotgun data profiling, taxon set analysis, and projection with public data. And finally, let's talk about quorum sensing. So biofilm quorum sensing is the regulation of genes in response to cell density, which can influence various functions. Quorum sensing does things like regulating genes involved in biofilm development, and also the sensing can promote biofilm dispersion. Here you can see software components that were specified for this workflow. We have things like Docker, Custom Script, Microbe Analyst. You can read through them, as well as QIIME2 and PyCrust2. And the programming languages that will be used are Python, RStudio, and Shell Script. So the submodules will have different learning objectives. For submodule 0, Concepts and Workflow Overview, the learning objective will be to receive fundamental concepts relating to microbiome and biofilm analysis. For submodule 1, Dataset Preparation and Quality Control, the learning objective will to be able to describe and manipulate dataset and toolkits that are relevant to the microbiome analysis project. Another learning objective in that submodule is to be able to perform a cloud-based workflow implementation. In submodule 2, the learning objectives will be to analyze a sequencing dataset and to map to relevant microbiome taxonomy. Also to retrieve and understand the microbiome community that's available on the input dataset, and to interpret microbiome analysis results from gold standard tools such as QIIME2. For submodule 3, or biomarker discovery, the learning objectives will be to run a metagenomics marker gene discovery tool such as PyCrust, and to predict and evaluate resulting genes, protein, and pathway markers from our use case dataset. And finally, for submodule 4, microbiome community and biofilm analysis, the learning objective will be to learn how to perform microbiome community analyses using PyCrust, Q2 PyCrust, BLAST, QS, a variety of different tools. So right here we can see how this will look. You can see the scientist will go through 1A, 1B, 1C, 1D, and 1E through all the different submodules using Jupyter Notebook, and there are given data sets that will help your workflow and assist with your learning. Right here, you can see an example of what a training module may look like. You can see there is the title where you see submodule 0 introduction. You can see the learning objective, and you will see blocks of code that can be ran and manipulated as you are going through. There are also quizzes to assess learning. So if you feel like you may not have learned something as adequately as you would have liked, you can always go back and try to learn it. And finally, at the end, after all of your learning, you should be able to get beautiful outputs like the ones shown with all the colors and information. We would like to acknowledge NIGMS pilots, Biomedical Research Cloud-based learning tools, and everybody else. This is funded by the South Dakota INBRE program. references, and I would like to thank you all for watching.